Okay, everyone. So welcome to this month's Heart Muscle Talks. My name is Perry Elliott, work at UCL, and we're really pleased today to have our guest speaker, Katia Gehmlich, who's going to be talking to us about Z-discs and their role in cardiomyopathy. Before I do that, I'd like to introduce my co-host today, Max Lorenzini, also from Barts. And Katia, it would be great if we can maybe begin by a little bit of background about yourself and what your current interests are, and then we can crack on with your presentation today, which we're looking forward to.
Sure. Thank you very much for the kind introduction. So I'm a basic scientist by training. Actually, even chemistry was my first degree. And this is why I'm going to give a kind of basic scientist perspective on cardiomyopathies today. I'm now based at the University of Birmingham, there since already 4 years. Time is going quickly. Before that was in Oxford, and before that at UCL, where I briefly overlapped with Perry and Cool. And do you want me to start sharing my screen?
Yes, please. That would be great. Thanks.
Cool. Just a moment while I get things up and running. And the laser pointer might be useful. Can you all see the screen?
That's perfect.
And you can hear me, okay, that's brilliant. So yeah, I'm going to talk about functional work on Z-disc proteins and their roles in cardiomyopathies. And again, kind of bear in mind, I'm a basic scientist, so this is my view on the world. I don't think I have to introduce much of what cardiomyopathies are to this audience. Just a short recap: hypertrophic cardiomyopathy, here shown on the left, kind of very stiff muscle, kind of reduced diastolic function, so impaired relaxation, but preserved systolic function most of the time. And then the other side of the spectrum, dilated cardiomyopathy, way worn out hearts, kind of poor pumping function, and quite often kind of candidates for transplant. And those are the two cardiomyopathies that are most relevant to the projects I'm going to talk about today. Cardiomyopathies, why are they important? I think everyone knows it's associated with a high risk of sudden cardiac death. And Christian Eriksen, who collapsed during the Euro 2020 match, is a prominent example. And I sometimes kind of make the joke, I work on football players dropping dead when people ask me what I do for a living. And what causes these sudden cardiac deaths is kind of ventricular arrhythmias that can be life-threatening. I'm also interested in a ability of the heart that is quite remarkable, and that is that the heart senses mechanical demand and can respond to it. It's a normal physiological process. It's called physiological cardiac hypertrophy, and it means that upon stimuli such as endurance training, pregnancy, or growth, the heart can meet the increased demand by increasing the cardiac output. And this is done in the heart because cardiac cells can't divide easily by increasing the size of each individual cell, a process called myocyte hypertrophy. And it's a complex network of signaling, not fully understood. And the reason why I'm interested in it is that I think genetic variants in proteins involved in these biomechanical stress signaling pathways can disturb signaling functions in the heart and thereby result in cardiomyopathies. I'm not saying this is causing all cardiomyopathies, but it's causing a subset of cardiomyopathies that are mechanistically quite interesting. And the Z-disc is one of the places where this sensing and signaling takes place. The Z-disc is a protein-rich structure that anchors the fin filaments, so the actin filaments of the contractile apparatus. And it's made out of many, many proteins. And a few of them have been shown to be disease genes for cardiomyopathies. And a couple of them are listed here. So titin, which is starting in the Z-disc, but it is much longer. But classical Z-disc proteins, filament C, alpha-actinin 2, myopalladin, muscle limb protein, teletonin, nebulate, and exelin. And today I'm going to focus on 2 or 3 of them. The first one I'm going to touch only briefly upon is filament C. It's a fairly recent disease gene for cardiomyopathy, so only I think in the last 10 years has become recognized as a disease gene for cardiomyopathies. And generally, truncations in filament C tend to cause dilated cardiomyopathy or arrhythmic cardiomyopathy, whereas missense variants tend to cause hypertrophic cardiomyopathy or restrictive phenotypes. Given the size of the protein and of the gene, it's quite a noisy gene in the sense that you will find lots of missense variants, but not all of them are pathogenic. And this is a dilemma clinical geneticists are facing. And here just taken an example of a filament C missense variant identified both in Glasgow by Caroline Coates and by Perry Elliott in London. Methionine at position 82 to lysine mutation, but currently classified as a variant of unknown significance, which means it's not actionable. Family screening is not recommended. But then if you look into biochemical data, and here localization in transfected cardiomyocytes, work by Matthias Gautel at King's College, you see definitely abnormal properties of this molecule. And this is basically where we need the basic science tools to trying to make sense out of variants that are currently classified as variants as of unknown significance. But this is all I'm going to talk about filament C today, even though we do a bit more work on this protein. Today I'm going to focus on 2 proteins, titin and alpha-actinin. Titin, the vast majority of variants described in titin are truncation variants, but there are a few missense variants, and I'm going to talk about one in the Z-disc portion of titin. And then the other project I'm going to talk about is alpha-actinin 2, a missense mutation causing hypertrophic cardiomyopathy. So you could boil down my activity to the question, how does a genetic variant cause cardiomyopathy? And there are different ways to address this question. One is to look at patient material, to look at mouse models, using in vitro experiments, and more we use genome-edited stem cells, iPS-derived cardiomyocytes. But today I'm only going to talk about mouse model and the combination of in vitro experiments, and I leave the cellular work for another day. So, and the first project, as I said, is about a titin missense variant. And it caused a particular phenotype, which I'm going to explain in a couple of minutes. But maybe to give a bit of a recap or introduction to titin. So titin is a bit of a beast of a protein. It's the largest protein known in nature, and it's pretty much 100 times bigger than an ordinary protein like GAPDH. It's expressed in the heart and skeletal muscle, where it has structure functions and signaling functions. There's extensive splicing going on, especially in the IBAT region. And being such a big protein, it also interacts with many other proteins. Because of its size, so 363 exons, in times of Sanger sequencing, it was pretty much impossible to look at titin and its role in cardiomyopathies. And only when next generation sequencing was rolled out into practice, titin became accessible as a gene to interrogate. And it was found a bit more than 10 years ago that up to 25% of familial dilated cardiomyopathy are caused by truncations in titin. And initially, this was met with quite a bit of skepticism because titin truncations were also found in normal populations at kind of low, but still quite high frequency, so 1 to 3%. And I think we now start to understand a bit better why this is the case, so why some variants are less penetrant. And this has something to do with that the ones in normal populations tend to be in exons that are not used in the adult cardiac transcript. the situation is far more difficult for missense variants in Titin, because a third of the population will carry a rare missense variant in Titin, but obviously not a third of the population is at risk for cardiomyopathy. So the challenge here is, how can we distinguish benign variants from disease-causing ones? And I think we're not there yet, but we are trying to address this question. And I think from this point, the missense variant I'm going to talk about is quite interesting. So we came across this not because we wanted a challenge and we wanted to work with the largest protein, but because of this family. And it was a family with a fairly unique phenotype. So on the one hand, kind of typical dilated cardiomyopathy, but on the other hand, individuals with prominent trabeculation that seem to develop with age, which I think is fairly unusual. And if you look at the pedigree of the family, it's typical autosomal dominant disease, quite a few affected individuals in the family, and 2 cousins were selected for whole genome sequencing to identify the underlying cause of disease in the family. But if you do this, so if you take 2 individuals and look which variants they have in common that are not in the reference genome, you end up with thousands or 10,000s of possibilities. So we then use the other affected individuals in the family for linkage analysis, basically to ask the question, which bit of a chromosome have all the ones who are affected by disease in common? And then whole genome sequencing can tell you which coding variants are on these bits of the chromosome. And this identified 2 changes. One was this missense change in Titin, but genetically equally valid was also a missense change in an enzyme called PDP2. But if you looked at biological functions, expression patterns, the money was on Titin. And because of that, we tried to characterize this missense variant, initially with in vitro experiments and then later with a mouse model. So where is this variant? So Titin, as I said, is a really large protein, and basically each single Titin molecule spans half a sarcomere from the Z disc to the M line. So kind of really long molecule. And the missense is in the second domain, so at the way N terminus, where in the so-called Z2 domain, an immunoglobulin domain, and together with Z1, it forms a complex with a protein called teletonin. And if you look closer into the structure, the variant is not at the direct binding interface, but in what is called a hydrophobic pocket. So basically hydrophobic residues stabilizing this beta shield for it. And if you introduce a charged residue as an aspartate, you would expect this structure to be quite affected by it. So that was the theory for modeling. We then expressed and purified the protein, and you could already see with kind of during the purification that the Titin mutant protein behaved very differently to the Titin wild type protein. So for example, the apparent size on a gel filtration column was much larger. And to follow this up more closely, we teamed up with structural biologists and performed something that is called small angle X-ray scattering, or SAX. And it basically can give you an idea about the size and the folding of a protein with fairly low resolution, but you can then use existing structure models to see how it fits and where it might deviate from the predicted structure. And I must admit, I don't fully understand these curves, but what you can clearly see, there is a striking difference between the wild type and the mutant protein in parameters measured. And that also argues for kind of partial unfolding of the protein. And then something I found a bit more intuitive is the predicted structure and size of the wild type protein is this, and this fits very nicely with the structural models derived from X-ray crystallography. Whereas the mutant protein seems to form these kind of partially unfolded aggregates and lumps of proteins together. So the idea is it is not folding properly. I mentioned earlier that the function of this part of titin is to bind to a protein called teletonin or TCAP. And what is remarkable about this interaction is it's the strongest non-covalent binding observed in nature. So you would think something that is strong is also important. So we probed for changes in this interaction in the presence of the mutation. And shown here in kind of more semi-quantitative assays, GST pull-down bindings, there is reduced binding to in the presence of the mutation compared to the wild-type protein. And to follow this up more closely, we used a biological phenomenon called fluorescence resonance energy transfer, or FRET. And the idea of this experiment is that if you bring two fluorescent proteins close to each other, and you bring them close to each other by linking each of them to a protein, for example, to titin and to teletonin, and if these two proteins bind each other, they bring these two fluorescent proteins really close to each other, and this allows a physical phenomenon called FRET. So light is basically transferred from one to the other. And shown here, this works really well with a pair of titin teletonin if it's titin wild type, and you get a strong FRET signal indicating interaction of both proteins. But if you introduce the mutation, you completely abolish this binding. So the take-home message from this in vitro experiments was that there is evidence of domain destabilization and protein misfolding, resulting in an overall bulkier conformation of the titin and definitely impaired binding to teletonin. But the issue is, while these in vitro experiments can suggest a pathogenic potential, it's way difficult to extrapolate from, looking at these tiny region of titin to the full length protein. And also, I think for titin missense variants at the moment, human genetics, like large families, are still required to evaluate these titin missense variants. So I think it's way difficult to base this on in vitro experiments alone. So in order to have confirmation that this missense variant is indeed pathogenic, but also to understand the mechanisms better, we generated a mouse model. And this is shown here. We introduced the coding changed at in the genome with CRISPR-Cas9 and could detect it at mRNA level and also at protein level. And then, to cut away long story short, the heterozygous mice are the situation of the patient. So you have one wild type allele, normal titin, and you have one titin allele carrying the missense change. But these mice didn't have a phenotype, not when they were young, not when they were old, not when you looked at molecular markers. And this seems to be the story of my life. I generate mice that don't have a phenotype. And this is partially because mice and humans are very different in their physiology. So mouse heart beats 10 times faster than a human heart. And there's also kind of behavior of mice and humans is very different. So, but what we can do if this happens is we can breed mice to homozygosity. So instead of having a normal copy and a mutated copy, we can generate mice that have both copies of titin mutated. And then you see evidence of mild dilated cardiomyopathy in these mice. So reduced systolic function and increased kind of ventricular dimensions. And also, if you look at molecular features, so you see an induction of the fetal gene program. This is a hallmark of cardiomyopathies. And you also see evidence of hypertrophic signaling, which again is a hallmark of cardiomyopathies. And then we also have other tools to provoke phenotypes, and they were used in this project. One is simply to age mice to at least a year. And the other one is to provide to give them chronic adrenergic stimulation. So we give them isoprenaline and phenylephrine for 2 weeks, and this causes pressure overload and hypertrophy. And this we use this now to replace something called transaortic constriction surgery or TAC surgery, because we feel it's a bit more gentle on the mice. It doesn't involve opening the chest and is also a bit quicker. And what you can see here with this adrenergic stimulation, you do get a robust response in left ventricular mass in both wild type and mutant mice, but only in the mutant mice there starts to be a decline of systolic function. And if you look at molecular parameters, again, the mutant mice have a much higher response to the challenge than the wild type mice undergoing the same challenge. So we then wanted to look into what's going on in these mice molecularly to understand the phenotype. So the first thing was to look at cellular dimensions, and this confirms the mice have hypertrophy, so the cells are longer, they are wider, and therefore have a larger area. We also looked at contractility, but maybe to our surprise, there wasn't much change in contractility of isolated unloaded cells. And also the way they did calcium handling was fairly normal. And obviously, if you mutate titin, you would think this could change the properties of titin. So we looked at all sorts of titin aspects. And the first one was to look at isoform composition of titin and titin amount in these hearts, and that was all fairly normal. And there's also a T2 band that some people say is indicative of titin turnover. And again, that looked fairly normal. So pretty much titin looked normal. And also at microscopy, the titin Z-disc epitope was where it should be. It colocalized nicely with other Z-disc proteins such as alpha-actinin. And if you looked on the other side, that's not shown now, on the M-band, the titin was also where you would expect it to be. Titin has been implicated in controlling passive stiffness of the heart. But passive stiffness in these hearts was way normal. The Z-discs looked normal under electron microscopy. And also trabeculation of these hearts was way normal using kind of high resolution microscopy. Some people say titin is also expressed in the skeletal muscle, so you may have a skeletal muscle defect, but skeletal muscle was way normal. So we basically didn't have any indication what was going on in these mice. And this is when you can start doing what we call fishing exercises or hypothesis generating approaches. And the first one we chose was RNA sequencing. And this was a bit disappointing because it resulted only in a very small number of differently expressed genes. And that's maybe something to do with the mild phenotype. But also the terms that came up were kind of fairly generic, proteasome and fibrosis. And if you looked at histology, there wasn't anything striking about fibrosis in these hearts. So that was all a bit disappointing. And this is why we went on to do a proteomics experiment using kind of the myofilament protein fractions, so where titin and actin and myosin and associated proteins are. And again, kind of we got way few hits, only 6, but these hits proved incredibly informative. And they basically helped us to develop a disease mechanism in these mice. The 2 downregulated proteins were teletonin and 4.5 limb domain 2. And upregulated were 3 heat shock proteins in a protein called MLF1, which again is a stress-induced protein. And here just shown the confirmation of reduction of 4.5 limb domain. It's happening at the protein level, and this could be part of the hypertrophic program, because whenever there is hypertrophic pathways activated, 4.5 limb domains 2 goes down. More interesting and more exciting was looking at teletonin. Teletonin, again, Western blot confirmed the dramatic downregulation of teletonin. And if you looked at microscopy, teletonin was completely lost from the Z-discs of these mice. But at transcript level, teletonin was there in normal amounts, which argues that everything that happens to teletonin happens at the protein level. So it's basically a protein destabilization. So we thought, great, we now have the cause of DCM in our mice because it's down to the lack of teletonin. So we looked at all sorts of aspects of teletonin biology, teletonin function, to find out which of these processes are affected by the lack of functional teletonin. The first thing is, it's published in the literature that phosphorylation of teletonin is really important for its function, but its phosphorylation was very normal. Then teletonin has been implicated in being essential for T-tubular organization, but T-tubers look very normal. And this has then been linked to calcium handling. So if the T-tubers are not properly functional, then calcium handling of the cells is affected. But again, we had very normal calcium handling. Again, another disappointment. Then teletonin had been implicated in apoptosis, but apoptosis pathways were not upregulated in our mice. The only thing we could observe is that the residual teletonin that was still left in our mutant mice is not in the myofilament, but accumulating in the cytoplasm. So there's a quite substantial increase of teletonin protein in the cytoplasm. And previous structure work has suggested teletonin that is not bound to titin is likely to be unfolded. So, and that brought us back to this hit of proteasome in the RNA-seq, to think maybe it is the teletonin that gets accumulated in the cytoplasm that is then marked for degradation by the ubiquitin proteasomal system. And indeed, we could find confirmation that teletonin is ubiquitinated. And another experiment could show that the proteasome is responsible for degrading teletonin in the mice. And this is shown here. So wild-type mice have nice teletonin striation in the heart. In the mutant mice, you lose the striation. But if you treat the mutant mice with a proteasomal inhibitor, here using epoxamycin, you do increase the protein levels again of teletonin, and you do get some striation back. I'm aware kind of a small screen is a bit hard to see this, so this is why it's kind of magnified once more. So striation in the wild-type mice, complete loss of this striated signal in the mutant mice, and then a partial restoration of the signal upon proteasomal inhibition, which tells us the proteasome is responsible for degrading teletonin. But at the same time, the proteasome doesn't seem to be able to clear it off completely. And this raised the question, if the proteasome cannot clear all of the unfolded teletonin, maybe it's not able to clear all the other proteins that would be the daytime job of the proteasome to do. And that can then lead to accumulation of proteins and to what is called a proteotoxic response. And indeed, here as an example for 1-year-old mice, there is induction of feature proteins and BAC3 related proteins and proteins. So there is indication of a proteotoxic response. And this is now just looking at one of these proteins, alpha beta crystallin. And if you focus on the last panel on the summaries, what does become apparent is that you need a challenge, either aging to a year or the chronic adrenergic stimulation, to really see this proteotoxic response, because it's not quite there in the young kind of mutant mice. So in conclusion, the mouse recapitulates aspects of the human disease, but not all of it. So for example, it doesn't recapitulate the tuberculation, and it only does it in the homozygous setting. Pretty much all titin functions are normal, apart from binding to teletonin. And because teletonin cannot bind to titin anymore, it's lost from the Z disks, it accumulates in the cytoplasm and leads to a proteasomal overload and a proteotoxic response. And this proteotoxic response becomes unmasked under challenge conditions such as aging the mice or kind of chronic adrenergic stimulation. And the interesting thing is, and I'm going to point this out later again, is that the proteasomal overload seems a common theme in cardiomyopathies. And this is just a brief graphical summary of these mice, kind of loss of teletonin, a proteasomal activation and activation of a proteotoxic response. Evidence, for example, in upregulation of heat shock proteins. Good, now moving on to another project, and that is looking into an alpha-actinin missense mutation causing hypertrophic cardiomyopathy. Alpha-actinin is a Z-dis protein. It is an antiparallel dimer and a rare cause of hypertrophic cardiomyopathy. And when I mean rare, there are so far only something like 7 variants identified. But the interesting thing is that more than half of them cluster in the actin binding domain of alpha-actinin. And this project focused on one of them, methionine at position 228 mutated to threonine. And it was initially identified in an Italian family, very large family, again, evidence of autosomal dominant disease. And the family here shown for the index patient had features of hypertrophic cardiomyopathy, so enlarged septum width, enlarged atria, but also very early onset atrial fibrillation. And the aim of the project was to generate a mouse model to gain insights into the disease mechanisms. So again, CRISPR-Cas9 used to introduce the mutation. And again, the story of my life, I generate mice that don't have an overt phenotype. So characterizing these mice with echocardiography at 3 months and at 9 months, they showed normal dimensions of the heart and normal function. And only when you start looking at molecular parameters, here again, the fetal gene program, you could see in male mice, but not in female mice, an induction of this fetal gene program indicative of cardiomyopathy. And the reason why these males might do this more is they are more active. These mice like to fight, the males, whereas the females sit there and get on with each other. And this increased activity level might have kind of provoked the phenotype in them. As I mentioned with the Titan project, what we normally do is, if we don't get a striking phenotype, we breed to homozygosity. But to our surprise, we didn't get any homozygote mice. And so we made our way backwards in embryonic development because it tells you they are embryonic lethal. And E15.5 was the last time point where we could reliably find homozygous animals and at Mendelian ratios. And that is roughly 2/3 through pregnancy. And we then performed a high resolution microscopic analysis of these hearts. And the first thing identified was in the mutant mice, ventricular septal defects in some of them, not seen in the wild type ones, and also changes in the chamber sizes, for example, especially the right ventricular chamber being enlarged and strangely in the left atrium being smaller. One thing when we prepared these samples that was obvious was that there was always residual blood in the embryonic mutant hearts, whereas the wild type hearts kind of were pretty clear of blood. And this is because after dissection, the heart still pumps a bit and thereby pumps out the blood and kind of gets flushed out. And the fact that this didn't happen in the mutant hearts suggested that they had poor contractility. So we found alpha-actinin is a sarcomeric protein. It's mutated, most likely the sarcomeres will look kind of really horrible and non-functional, but they looked surprisingly good. So the homozygous had alpha-actinin at the Z-dis striation where it should be co-localizing with Titan. And only when starting to do quantitative analyses, it became apparent that there was a bit less co-localization with Z-dis Titan. Another thing observed was increased sarcomere length and a reduced number of nuclei. If we zoom in at electron microscopy level, the width was normal, but the distribution was wider, and there were also these kind of irregular Z disks, so they weren't as nicely formed. And one side observation was that E15.5 is an interesting one in the sense it's a time when heart cells can still divide. And it's interesting in the sense, kind of, it's fully functional contractile cardiomyocytes with sarcomeric apparatus here shown with the striation. And in order to divide, these cells need to disassemble the sarcomeric apparatus completely, then divide, and then reassemble the sarcomeric apparatus. And we had individual cells in the mutant mice that clearly didn't make this process normally. So they were abnormal. And this prompted us to look at the cell cycle. And here, first analysis by qPCR, kind of a dysregulation of cell cycle transcripts at various stages. And we also quantified the number of phosphohistone positive cells, so the number of cells basically going through the cell cycle, and that was reduced in the mutant hearts. And then if you zoom in more closely into these dividing hearts, what you can see for the wild types is that they really disassemble the contractile apparatus, whereas the mutant hearts seem to have residual alpha-actinin left in cells trying to divide. And I think this might stop them progressing through the whole cell division because they fail to disassemble these sarcomeres completely. Again, in order to understand more about the mechanisms behind, we performed a proteomics experiment, this time observed a couple of more hits. And if you look into all the downregulated proteins, there was one common theme and one pathway that really stuck out, and that was mitochondrial metabolism. So we think maybe these hearts also die, or these mice embryos die, because their hearts don't have enough energy for contraction. The upregulated proteins gave a bit more of a complex picture, but terms like protein translation, folding, and proteasome popped up. And then another thing you can do with proteomics experiments is you can obviously look very detailed in a protein of interest. And we did this for alpha-actinin, and proteomics suggested a striking downregulation of the protein. At transcript level, interestingly, it was upregulated. But if you looked at protein level on Western blot, this validated the proteomics findings, really saying it's less than half of the protein left. And that again, kind of with proteasome as a hit in the proteomics experiments, suggests degradation by the ubiquitin proteasomal system. And we think the upregulation of the transcripts is a desperate measure of the cells trying to increase the amount of alpha-actinin. So in summary of this project, the mature or the adult mice didn't tell us much about the disease mechanisms. They more gave us confirmation that this variant is indeed causing cardiomyopathy, whereas the homozygous embryos kind of gave more insights, and they identified that the protein destabilization of alpha-actinin contributes to the phenotype, potentially also mitochondrial defects. And there's now the thinking that this might actually be a direct link between Z-dis proteins and mitochondrial function. And also it identified maybe a quite surprising role of alpha-actinin in controlling the cell cycle. So, and coming to the end of my talk, I hope I could convince you that functional studies can help us to confirm the pathogenicity of a variant in kind of whole organ models, but they can also tell you that mice are not humans, and not everything seen in the human patients can be modeled or kind of found in the mice. And this is partially because cardiomyopathies tend to be late onset in humans, and mouse have a way different physiology. Nevertheless, the mouse model can provide insights into disease mechanisms. And one thing that keeps, for example, coming up is the role of protein quality control and protein turnover in cardiomyopathies. And they can even help to uncover new biological functions of proteins, like the role of alpha-actinin in cell cycling. And I want to mention once more, in parallel to the mouse models, there is now kind of a real strong potential for iPS cardiomyocytes to be used as cellular models. So kind of with the hope of being able to do human cellular models, but also to be able to use less mice for our research. And we are doing this in parallel to our mouse models, but I've not talked about it today. And yeah, at the end of the talk, I would like to acknowledge all the people who contribute to the work. So my group in Oxford back then, and then now kind of taken over by the group in Birmingham, and also many collaborators, both kind of basic scientists, microscopists, clinicians, and funding from the Wellcome Trust, the Medical Research Council, and the British Heart Foundation. And I thank you for your attention and very happy to take questions.
Katia, thank you very much. Huge amount of work. And I think you've spanned sort of the spectrum, I suppose, from very subtle to very severe in the kind of phenotypes you might expect to see with these two different models. We're open to questions from the audience, so please, people, post your questions on the chat, or raise your hand if you want to join in the conversation. Just as a reflection, I mean, you've gone into tiger country with tightened missenses because obviously they're really common, and it's something that we and others are interested in, because I think it's hard to make the case they're an important contributor by themselves to disease, but as a predisposing mechanism, perhaps in combination with other genetic variants, or as you've shown, in combination with other stressors. And I think it's an emerging new paradigm.
Yeah, it's difficult because obviously you cannot do a mouse model for each tightened missense you identify. But the idea, for example, is, and we kind of proposed this, if you have a mouse model for a region of titin and you understand the biology of this region very well, if you find then other missense variants that in biochemical experiments gave you a similar feature. So for example, if you were to identify one that also reduces binding to teletonin or gives a structural destabilization in this region, you wouldn't need to do another mouse model because you already have the insight into the biology. So maybe we don't need to make 30,000 mouse models. We only need to make 100 for all the different functional regions, and then can use those. Still a massive amount of work.
Huge amount of work, yeah. And also I'm struck by because the phenotypes are subtle, and as you pointed out, I mean, you were disappointed by the lack of a signal in many of the functional assays that you did, but you eventually sort of struck upon those where you clearly had a consistent signal. But it means that the range of functional analyses that you need to do is really quite big, right? For some of these variants, if the effect.
You could argue if you looked at the right one
first of all,
you could have, I could have saved myself a lot of work. But I think this is the advantage of now knowing the biology of this region, that the next ones it would be much easier if it's all about titin teletonin interaction in this region. And you would think kind of this seems to be the prime property of the Z1, Z2 is to kind of keep teletonin there.
Yeah, Max, sorry, I interrupt you.
No, but you didn't. But from a very, very clinical perspective, what strikes me from your titin family, and the only other one that I've come across that was recently published by Pablo Garcia and his group, which was a very, very large family, they both have a lot of affected members, which sort of is hard to reconcile with variants that are so common in the general population and have a low, or should have a low penetrance in response to stressors. So I find it hard to tie it all together.
Yeah, I mean, we were maybe lucky with this particular one because it has quite a striking phenotype in the family. And the way this was done was basically families were selected where candidate gene approaches hadn't identified a cause of the cardiomyopathy, but the cardiomyopathy was striking and also quite unique. So, and therefore it was clear it has to be something genetic. And that was, for example, this family was selected before Titin was part of gene panels.
You had a second hit in that family, though, didn't you? And then I forget the particular enzyme. Did the two co-segregate each other?
They completely co-segregated in the pedigree. And this is why we took the Titin missense out of its context, put it into a mouse model, and said if we then see a phenotype, the enzyme doesn't contribute. And it was basically looking at the biological functions. So the enzyme just is expressed in all sorts of tissues, didn't have any cardiac specific phenotype. So,
but could it be the combination of the two, though? I mean, it's this point again that
it could be. It could be. We cannot exclude it. And I was surprised that they were genetically equally valid. They had the same weight and scores. And it's only based on biological function that I thought it's a Titin.
I mean, because probably one of the best models for Titin missense disease is recessive, so skeletal myopathy. The Atlas Mountain thing. I think, now that we're looking for it, you get the hint in some families with particularly dilated cardiomyopathy that the second hit is important in terms of modulating the phenotype. I mean, I know we've got Eloisa on, or we did have Eloisa on the line. I don't know, Eloisa, do you want to express profound doubt or support for the Titin missense?
You should not ask this question. It's not the pathogenesis of the hypothesis or the mechanism of disease, but the problems we have in clinics is the lack of segregation and, unfortunately, the lack of clinical evidence that we are observing a true disease-causing effect. Vice versa, I had a question about the Titin alanine 178, I guess, aspartic acid variant, right? And supposed to have a teletonin variant in the domain of Titin, in which teletonin is abnormal, but as a consequence of the Titin defect. Do you expect a similar effect, a similar damage, or not? Rather than having Titin, having a teletonin.
I know what you mean. So you'd suggest if we had a teletonin variant that doesn't
wild type Titin.
Yeah, that doesnn't bind to Titin anymore.
That given domain. Do you expect a similar effect?
I would expect it. I don't know. I know teletonin variants do exist, but they are incredibly rare. The interesting thing with teletonin is if you knock it out in a mouse, the mouse is absolutely fine until again you start challenging it. So this is quite puzzling that I always think kind of if the interaction between two proteins is designed to be incredibly strong at force level, you would think it's really important, and then you take one of the components away and the mice are pretty much fine until you really kind of challenge them. So but to answer your question, I don't know whether there are any teletonin mutations doing exactly the same. If they were identified, I would think they do exactly the same mechanism, but I'm not 100% sure.
Because the main issue that you have raised in this, irrespective of the two examples presented in your lecture, is the question of protein-to-protein interactions, which is far more complex, than expected. In particular, when we investigate this problem in humans and in human samples, and in particular when we want to look at 2 or 3 proteins expressed in the same heart, for example, of a patient with 1 genetic defect, not necessarily 3 genetic defects. What I mean is the protein-to-protein interaction is really important to be the future scope in research, but very difficult to explore in vivo in humans and in heart samples, unless there's some, I don't know, fantastic new ideas on how to do. I think that generating an animal model for each mutation is impossible, of course.
No. and
yeah, you've said it before, I fully agree. But we are still, we are not progressing very much. We are still, in a position in which all of us conclude our studies by saying we need future studies, we need functional investigation in order to assess blah, blah, blah. So it means that something, a revolution tool, some revolutionary tool should in some way be considered for the future. Don't you think that the mice model available now are not enough to start searching for tools rather than searching for mechanism of single mutations? Tools for exploring protein-to-protein interactions in vivo.
So I think the mice are better for
you have many mice
mechanistic insights, but the issue is it takes. I mean, the Titan project took 7 years, and that's not, in the clinical time frame. This is not what you want to wait 7 years until a basic scientist comes back and says, kind of, we think now it's pathogenic, or we sit on the fence. So I think there's a lot of potential with the iPS cardiomyocytes, genome editing. So I think we have now tools we didn't have 10 years ago. And I think they might help us to progress. And one example, again, is the filament C missense variant I showed in the very beginning. Currently clinically characterized as a variant of unknown significance, which means we cannot do genetic testing in family members and similar stuff. But maybe using cellular models might tell us quite quickly that in a cardiomyocyte, something is wrong in the presence of this variant.
Katia, I think Alex would probably tell us that artificial intelligence is the way forward here, because you've got a model at scale, right? So some of this stuff may have to be inferred or developed in silico, based
and
upon.
And there is, in terms of artificial intelligence, there is now AlphaFold, for example. So AlphaFold is a structure basically you can now predict structures of a protein sequence you couldn't do 5 years ago. And I think alpha missense is the development coming from this. So I think, yes, artificial intelligence predictions have their role, but I think we still need, I'm vouching for my own job, we still need basic scientists in a wet lab to find out about biological functions. So
it may be that the biological functions work in tandem with this in silico modeling.
I think both together is really powerful.
Yeah, I mean, your point about doing the right experiment, rather than sort of going through all the traditional ones, it may be that artificial intelligence can guide you towards the right experiments to get maybe a quicker answer, possibly.
Yeah.
But do you expect some change in peripheral biomarkers? Are you testing peripheral biomarkers in these animals, or do you think that is useless?
So we haven't looked at this. It's hard to. hard to think what the biomarkers should be that are changed for these particular mechanisms or pathways.
I also think, Eloise, I mean, these phenotypes, they're really subtle until you sort of stress the system. I mean, for me, this you highlighted the proteasomal system, but this is the recurrent story now, isn't it, in heart muscle disease? It's this predisposition. It's very, for me, it's very analogous to ion channel disease, in that you can have a highly penetrant variant which gives you long QT syndrome, or you can have a collection of variants which, in combination with some environmental perturbations, pushes you over the threshold at which you get membrane instability sufficient to give you an arrhythmia. I think we're seeing the same in heart muscle disease. It's why do we keep seeing the same proteins implicated in GWAS studies and so on? It's titin, you know, it's BAG3. It's.
I'm sorry. Are you saying that it's a polygenic issue?
No, well, polygenics are part of it, but it's setting the not the thermostat, whatever it is. It's some patients are over the threshold and they have overt disease. Others need something else to push them over, and titin is a great model for that, because it could be hemodynamic overload as in pregnancy, or chemotherapy, or alcoholism. But the person is predisposed. Now, are missense variants alone sufficient? Time will tell. But it may be a missense in a titin plus some missenses in BAG3 plus something else will also give you a phenotype. But I think that resilience hypothesis to the myocardium, I think, is definitely one of the models that's emerging. Yeah, these final common pathways, including, as you said, the
protein turnover, protein control. Whichever model I start, I end up with the same conclusion, which is sometimes a bit kind of, no, not again. Cool. There is a question in the chat about decreased actin and protein expression levels. The question is it possible that the actinin 2 reduction could affect myofibrillogenesis, so the formation of the...
Interesting. I haven't got that. You're getting secret questions, were they?
It's in the question and answers. And it was from Maicon Landim Vieira. So interestingly,
we have a really short answer, Katia, because we've got 1 minute left.
Yeah, sure. So and as I said, we start doing a cellular model, and yes, there we have evidence that the generation of sarcomeres is impaired. Strangely, in the mouse model, the sarcomeres look fairly normal, which was a surprise. So it's not like other mouse models where they do get stuck in making mature sarcomeres. But again, the cells can tell you something else, because they are compatible with function that a mouse heart wouldn't be compatible with. Cool. Yeah, okay. I'm aware I'm between everyone and Pap are going home, having a weekend.
Katia, I mean, thanks for that. I mean, I say really great work, and
thanks for having me.
Thank you for coming. I'd like to thank everybody for attending, and we look forward to seeing everybody next time. Thank you, Max, as well.
Thank you.
Bye-bye.
Bye-bye.
Bye-bye now.